In the depths of a dimly lit laboratory at King's College London, a young woman peers through the eyepiece of an X-ray machine. She watches as invisible beams pierce through crystallized strands of the most mysterious substance on Earth, DNA, the very blueprint of life that had puzzled scientists for decades. What would drive a brilliant chemist to spend years in basement laboratories, exposed to dangerous radiation, capturing shadows of molecules too small to see? How would a single photograph, emerging from a chemical bath like a ghost from darkness, unlock the secret that had eluded the greatest minds of the 20th century? What forces would conspire to deny her the recognition she deserved, even as her work transformed our understanding of life itself? And how would her meticulous measurements, recorded in neat handwriting by candlelight, ultimately give humanity the power to read, edit, and rewrite the very code of existence? Meet Rosalind Franklin, the woman whose steady hands and unwavering scientific rigor would capture the first clear photograph of DNA's structure, revealing secrets that had been locked away since the dawn of life itself. Her story begins not in a laboratory, but in a London nursery, where a young girl's insatiable curiosity about the invisible forces governing nature would set her on a collision course with one of science's greatest mysteries. As we descend into the basement laboratories where X-rays would illuminate both the architecture of life and the courage of one woman who refused to accept anything less than truth, we discover a tale of dedication, controversy, and discovery that echoes through every strand of DNA in every living cell on Earth. The journey into Rosalind Franklin's world begins now, in a time when women weren't supposed to unlock nature's deepest secrets. Yet one woman's determination would prove that the molecules of life reveal their structures not to those who demand quick answers, but to those patient enough to listen to what shadows have to say. Born into a prosperous London family in 1920, young Rosalind Franklin displays an insatiable hunger for understanding the invisible forces that govern the natural world around her. While other children play with dolls and toy soldiers, she constructs elaborate experiments in her nursery, mixing household substances to observe their reactions, recording each result in notebooks with the precision of a seasoned researcher. The maids whisper about finding her asleep at her desk, surrounded by diagrams of crystals she's drawn from memory, after visiting the Natural History Museum. Her father, a merchant banker, watches with growing concern as his daughter abandons traditional feminine pursuits, instead losing herself for hours in chemistry sets and scientific journals. Ellis Franklin had envisioned his daughter hosting elegant tea parties and mastering the social graces that would secure her a prosperous marriage. Instead, he finds her in the garden shed, conducting experiments with acids and bases, her dress stained with chemicals, her eyes bright with discovery. When guests visit their Notting Hill home, Rosalind retreats to her makeshift laboratory rather than practice piano or embroidery, much to her mother's quiet dismay. At St. Paul's Girls' School, Franklin's teachers recognize something extraordinary, a mind that refuses to accept simple explanations, always digging deeper into the fundamental questions of matter and energy. Her chemistry teacher, Miss O'Connor, recalls finding the young student after hours in the laboratory, repeating experiments until she understood not just what happened, but why. Franklin's questions often leave her instructors scrambling for answers. Why do crystals form in specific patterns? What forces hold atoms together? How can we see things too small for any microscope? Her relentless curiosity transforms every lesson into an exploration of nature's deepest mysteries. The teenage Franklin makes a declaration that shocks her family. She will dedicate her life to science, even if it means sacrificing the conventional path of marriage and motherhood expected of women in 1930s Britain. At the dinner table one evening, as her parents discuss potential suitors and social engagements, 16-year-old Rosalind announces her intention to become a scientist. The silence that follows feels heavy as lead. Her father's face darkens while her mother's hands tremble slightly as she sets down her teacup. In a society where women's colleges at Cambridge don't even grant full degrees, where female scientists work in the shadows of their male colleagues, Rosalind's declaration sounds like social suicide, yet she remains unmoved, her determination as crystalline as the structures she longs to study. Cambridge University becomes her sanctuary, where she immerses herself in the emerging field of physical chemistry, learning to see the world through the lens of molecular structures and atomic bonds. The Gothic spires and ancient courtyards fade into background as Franklin spends 18-hour days in laboratories and libraries, absorbing knowledge like a sponge. She finds kindred spirits among the few other women pursuing science, forming study groups that meet in dormitory rooms where equations cover every available surface. Her professors note her exceptional ability to visualize three-dimensional molecular structures, a skill that will prove invaluable in her future work. The mathematical beauty of chemistry captivates her. How simple rules govern the dance of electrons. How molecular geometry determines a substance's properties. How the invisible architecture of matter shapes the visible world. World War II erupts around her studies, but Franklin remains focused on her research into the microstructure of coal, work that will later prove crucial to Britain's war effort. While bombs fall on London and her male colleagues enlist, Franklin channels her patriotic duty through science. Her investigation into coal's permeability helps develop better gas masks, potentially saving thousands of lives. The work requires her to master new techniques, studying how different types of coal absorb and release gases, mapping their internal structures through painstaking experimentation. She works through air raid sirens, refusing to abandon her samples even as others flee to shelters. The war years forge her scientific discipline. Every experiment must count. Every material must be conserved. Every finding might save lives. Her doctoral thesis on coal permeability earns recognition, but Franklin knows this is only the beginning. She dreams of unlocking the secrets of biological molecules, the building blocks of life itself. The microscopic pores in coal fascinate her, but she yearns to study structures with even greater significance. Late nights in the Cambridge Library, she reads everything available about proteins, nucleic acids, and the mysterious substances that carry hereditary information. The emerging field of molecular biology calls to her like a siren song. She begins to see connections between her work on coal and the larger questions of biological structure. If she can map the invisible architecture of carbon, perhaps she can do the same for the molecules that define life itself. A fellowship in Paris opens new doors, where she masters X-ray crystallography under the guidance of Jacques Mering, learning to capture the invisible architecture of crystals through the ghostly shadows they cast. The Laboratoire Central des Services Chimiques de l'État becomes her new home, its sophisticated equipment far surpassing anything available in war-torn Britain. Mering, a master crystallographer, recognizes Franklin's exceptional spatial reasoning and mathematical precision. He teaches her to read diffraction patterns like musical scores, each spot and shadow revealing information about atomic arrangements. The technique requires an unusual combination of experimental skill and theoretical understanding, preparing perfect crystal samples, precisely controlling X-ray beams, then interpreting the resulting patterns through complex mathematical analysis. In the laboratories of the French capital, Franklin discovers her true calling, using X-rays to peer into the heart of matter, revealing structures too small for any microscope to see. The process becomes almost meditative for her. In the darkened X-ray room, she positions crystals with the delicacy of a jeweler setting diamonds. The invisible radiation passes through her samples, bending and scattering according to the arrangement of atoms within. Hours later, developing the photographic plates, she watches patterns emerge from the chemical baths, abstract designs that encode nature's architectural blueprints. Each successful photograph feels like receiving a message from the molecular world, written in a language only she and a handful of others can read. The technique becomes second nature to her, positioning tiny crystals in the path of focused X-ray beams, then studying the diffraction patterns that emerge like fingerprints of molecular structure. Franklin develops an intuitive understanding of how different crystal orientations produce different patterns, how exposure times affect image clarity, how temperature and humidity influence results. She fills notebooks with observations, creating a personal encyclopedia of crystallographic knowledge. Her French colleagues marvel at her ability to coax clear patterns from difficult samples, to spot subtle features others miss, to extract maximum information from every experiment. The precision required suits her perfectionist nature. A fraction of a degree's misalignment can ruin hours of work. Her work on carbon structures gains international attention, but Franklin feels the pull of an even greater mystery: the biological molecules that carry the instructions for all living things. At scientific conferences, she hears whispers about DNA, the substance somehow responsible for heredity. Scientists know its chemical composition, but not its structure. How does this molecule store information? How does it replicate itself with such fidelity? The questions consume her thoughts during long walks along the Seine. She begins reading everything available about nucleic acids, teaching herself the biochemistry needed to understand these complex molecules. The challenge seems almost impossible. Biological molecules are far more complex than the simple crystals she's studied. Their structures irregular and difficult to crystallize. Letters from London arrive with an offer that will change everything: a position at King's College to investigate the structure of DNA, the enigmatic substance that somehow contains the secret of heredity. Professor John Randall's invitation promises state-of-the-art equipment and the freedom to pursue the most important question in biology. Franklin hesitates, comfortable in Paris, where she's built a reputation and found acceptance, but the opportunity to work on DNA proves irresistible. She knows the competition will be fierce. Teams around the world are racing to solve this puzzle. Yet she also knows her unique combination of crystallographic expertise and meticulous methodology gives her a real chance at success. Franklin accepts the challenge, unaware that she's about to embark on the most important and controversial work of her career: research that will unlock one of biology's greatest mysteries. Her Parisian colleagues throw a farewell party, presenting her with a silver compass to help navigate the mysteries of life. She packs her notebooks, her most successful diffraction photographs, and the hard-won expertise of 4 years in France. The Channel crossing feels symbolic, leaving the known world of carbon crystals for the uncharted territory of biological molecules. London appears gray and austere after Paris, but Franklin's excitement overshadows any apprehension. She arrives at King's College in 1951, carrying with her the skills and determination needed to solve a puzzle that has confounded scientists for generations. The biophysics unit occupies cramped basement laboratories, a far cry from the elegant Parisian facilities. Yet Franklin sees potential in the specialized X-ray equipment, particularly a new microcamera that can capture fine details previous instruments missed. She meets Maurice Wilkins, who's been working on DNA but seems eager for collaboration. The initial interactions are cordial, both scientists discussing the technical challenges of working with biological samples. Franklin begins setting up her laboratory space, arranging equipment with the precision of a surgeon preparing an operating theater. The laboratory becomes her second home, filled with the hum of X-ray equipment and the chemical smell of developing solutions that will capture history's most important molecular portrait. She establishes routines that will govern the next 2 years of her life, arriving before dawn to check overnight exposures, spending hours in the darkroom developing plates, analyzing patterns until her eyes blur from strain. The basement windows offer no natural light, creating a timeless environment where day and night lose meaning. Franklin brings a cot for the nights when experiments run too late to justify going home. She learns every quirk of the equipment, every variable that might affect her results. Franklin begins her systematic study of DNA fibers, adjusting humidity levels and X-ray exposure times with the precision of a master craftsman perfecting their art. Unlike other researchers who rush to test theories, she commits to mapping DNA's behavior under every possible condition. She constructs humidity chambers that can maintain moisture levels within fractions of a percent, discovering that DNA's structure changes dramatically with water content. The fibers prove frustratingly difficult to work with. Too dry and they produce unclear patterns. Too wet and they dissolve. Finding the perfect balance requires endless patience and countless failed attempts. Each experiment brings new insights into DNA's behavior, revealing that the molecule can exist in different forms depending on the moisture content of its environment. Franklin designates these as the A and B forms, each producing distinct diffraction patterns. The A form appears at lower humidity, creating complex patterns that suggest a highly organized structure. The B form emerges at higher moisture levels, its patterns simpler but somehow more revealing. She realizes that understanding both forms will be crucial to solving DNA's structure. The discovery excites her. Previous researchers had seen confusing, contradictory patterns because they hadn't recognized they were looking at two different configurations of the same molecule. Her meticulous notes record every detail, building a comprehensive picture of DNA's properties through hundreds of careful observations and measurements. Franklin's laboratory notebooks become legendary among those who later study them. Every experiment documented with exhaustive detail, every calculation checked and rechecked, every observation recorded no matter how seemingly insignificant. She creates standardized procedures for preparing DNA fibers, controlling experimental conditions, and analyzing results. The systematic approach stands in stark contrast to the more intuitive methods favored by some competitors. Where others might follow hunches or build speculative models, Franklin lets the data speak for itself, accumulating evidence like a detective building an airtight case. The patterns begin to whisper their secrets, suggesting a structure of elegant simplicity hidden within DNA's complex chemistry. As she prepares yet another sample, adjusting the humidity chamber with practiced precision, Franklin senses she's approaching something momentous, though the full picture remains tantalizingly just beyond reach. The X-ray crystallography laboratory becomes Franklin's domain, where she works long hours in near solitude, coaxing DNA fibers into perfect crystalline arrangements for photography. The basement rooms at King's College feel like a submarine, windowless, isolated, filled with the constant hum of electrical equipment. Franklin arrives before sunrise, her footsteps echoing in empty corridors as she descends to her underground realm. The DNA samples demand constant attention, their delicate fibers requiring precise environmental control. She constructs specialized chambers where temperature and humidity can be adjusted by fractions of degrees. Each fiber must be mounted on tiny frames, positioned with the care of a watchmaker assembling microscopic gears. The work requires steady hands and infinite patience. A single tremor can destroy hours of preparation. Franklin develops a rhythm, her movements becoming almost meditative as she prepares sample after sample, seeking the perfect specimen that will reveal DNA's secrets. Her colleague, Maurice Wilkins, initially welcomes her arrival, but tensions soon emerge as Franklin establishes her independence, refusing to be treated as a mere assistant in the DNA research. The misunderstanding begins immediately. Wilkins assumes she's been hired to help with his project, while Franklin believes she's been given her own independent research program. Their first meetings crackle with unspoken conflict as Franklin sets up her equipment without consulting him, choosing her own experimental approaches. Wilkins, accustomed to the collaborative atmosphere among male scientists, finds Franklin's professional boundaries incomprehensible. She declines invitations to informal discussions at the local pub, preferring to analyze data alone in her office. When he suggests experimental directions, she listens politely, but follows her own methodical plan. The cultural clash deepens. In 1950s Britain, female scientists are expected to be helpmates, not independent researchers. Franklin's insistence on autonomy strikes Wilkins as arrogance rather than professionalism. The delicate process of preparing DNA samples requires infinite patience, extracting pure fibers, controlling humidity to precise levels, then positioning them perfectly in the X-ray beam's path. Franklin sources DNA from calf thymus glands, carefully extracting the genetic material through chemical purification. The resulting fibers resemble spider silk, nearly invisible to the naked eye. She develops techniques for drawing these fibers into uniform strands, using glass rods and steady hands to create samples thin enough for X-rays to penetrate. The humidity control proves crucial. Too dry and the DNA becomes brittle, too moist and it dissolves into useless gel. Franklin constructs sealed chambers with salt solutions that maintain specific humidity levels, learning through trial and error which conditions produce the best diffraction patterns. Each sample takes hours to prepare, and many fail before ever reaching the X-ray apparatus. Franklin develops new techniques for maintaining DNA in its different structural forms, discovering that the molecule transforms dramatically as moisture levels change around it. Through systematic experimentation, she maps the transformation between DNA's configurations. At 75% humidity, the fibers adopt one structure. At 95%, they shift to another. The discovery excites her. Previous researchers had obtained confusing results because they hadn't recognized these distinct forms. Franklin designates them A and B, creating standardized protocols for maintaining each configuration. She fills notebooks with observations about how DNA behaves under different conditions, noting color changes, flexibility, and crystalline properties. The work requires her to become part chemist, part physicist, part engineer, designing equipment, mixing solutions, and calculating molecular behavior. Her laboratory becomes a controlled environment where every variable can be measured and adjusted. Day after day, she adjusts the complex X-ray equipment, fine-tuning beam intensity and exposure, times to capture the clearest possible diffraction patterns from DNA crystals. The X-ray camera resembles a small cannon, its copper target bombarded with electrons to produce radiation. Franklin learns every quirk of the machine, how temperature affects beam stability, how voltage fluctuations blur images, how tiny misalignments produce worthless photographs. She develops an intuitive sense for the equipment, detecting problems through subtle changes in sound or vibration. Exposure times vary from hours to days, requiring careful planning to maximize each experiment. Franklin creates detailed logs tracking every parameter: beam intensity, sample orientation, exposure duration, development conditions. The systematic approach allows her to reproduce successful results and learn from failures. She modifies the equipment, adding cooling systems and stabilizers that improve image quality. The darkroom becomes her sanctuary, where she develops photographic plates in red-lit silence, watching as ghostly patterns emerge from the chemical baths like messages from the molecular world. The ritual soothes her, mixing fresh developer, maintaining precise temperatures, timing each step with metronomic precision. Franklin works alone in the crimson darkness, her movements practiced and efficient. The X-ray films float in their chemical baths, slowly revealing the diffraction patterns captured during long exposures. She watches dots and arcs materialize on the emulsion, each one encoding information about atomic positions within DNA. The best photographs show clear, symmetrical patterns that hint at underlying order. Franklin develops an aesthetic sense for quality diffraction patterns, recognizing immediately which images contain useful data. The darkroom offers respite from laboratory tensions, a space where only she and the molecules communicate. Each photograph tells part of DNA's story. The spacing of spots reveals the distance between atomic layers, while their intensity indicates the density of matter at different positions. Franklin becomes fluent in the language of diffraction patterns, reading the abstract images like musical scores. A spot's position encodes the spacing between repeating structures. Its brightness reveals how many atoms contribute to that reflection. She measures angles with protractors, calculates spacings through trigonometry, builds three-dimensional models in her mind from two-dimensional shadows. The mathematics involved would intimidate most biologists, but Franklin's physics training serves her well. She derives equations relating diffraction patterns to molecular structure, checking her work against known crystals before applying the techniques to DNA. Each successful calculation brings her closer to understanding the molecule's architecture. Franklin's systematic approach sets her apart from other researchers. Where others might rush to publish preliminary findings, she insists on gathering comprehensive data before drawing conclusions. The scientific community buzzes with DNA speculation, pressure mounting to be first with the answer. Yet Franklin refuses to be hurried. She knows that extraordinary claims require extraordinary evidence. While competitors build speculative models based on chemical theory, she accumulates experimental facts. Her notebooks grow thick with measurements, calculations, and observations. She photographs DNA under every conceivable condition, building a comprehensive database of its behavior. Critics whisper that she's too cautious, too slow, missing the forest for the trees, but Franklin believes that only through complete understanding can truth emerge. She's seen too many premature theories collapse when confronted with contradictory data. The relationship with Wilkins deteriorates as professional disagreements escalate into personal conflicts, creating a hostile atmosphere that makes Franklin's already difficult work even more challenging. Communication breaks down completely. They avoid each other in corridors, schedule equipment use to prevent overlap, communicate through terse notes when absolutely necessary. Wilkins complains to colleagues about Franklin's difficult personality, while she confides to friends about his patronizing attitude. The situation becomes known throughout King's College, creating camps of loyalty that further poison the atmosphere. Franklin finds herself increasingly isolated, excluded from informal scientific discussions where ideas flow freely. The loneliness weighs on her, but pride prevents reconciliation. She channels frustration into work, spending even longer hours in the laboratory, determined to prove her worth through results rather than politics. Despite the interpersonal tensions, Franklin continues her methodical investigation, accumulating hundreds of X-ray photographs that gradually reveal DNA's hidden architecture. Her filing system grows elaborate, photographs organized by date, sample type, humidity level, exposure conditions. She develops a visual memory for patterns, able to recall specific images from months earlier. The collection represents thousands of hours of work, each photograph a hard-won piece of the puzzle. Franklin begins to see recurring features across different samples: consistent spacings, repeated angles, symmetrical arrangements. The evidence accumulates like sediment, slowly building toward undeniable conclusions. She resists the temptation to leap ahead, instead letting the data guide her understanding. The photographs whisper their secrets gradually, revealing DNA's structure through patient observation rather than brilliant deduction. She identifies two distinct forms of DNA, the dry A form and the wet B form, each with its own characteristic diffraction pattern and molecular arrangement. The A form produces complex patterns with many reflections, suggesting a tightly packed structure. Its photographs show intricate designs that challenge analysis, beautiful but bewildering. The B form yields simpler, more elegant patterns, clear spots arranged in distinctive crosses and diamonds. Franklin realizes that understanding both forms is crucial. The DNA's ability to transform between configurations must relate to its biological function. She develops techniques for capturing each form at its most revealing, learning which humidity levels produce the sharpest patterns. The dual nature of DNA fascinates her. How can one molecule adopt such different configurations? The answer, she suspects, lies in water's interaction with the molecular structure. The B form proves particularly intriguing, its X-ray pattern suggesting a highly regular repeating structure that could hold the key to understanding DNA's biological function. Franklin focuses increasingly on these samples, perfecting conditions that maintain the B configuration. The patterns show remarkable regularity, spots spaced with mathematical precision, intensities that follow predictable patterns. She begins to see helical signatures in the data, spiral staircases revealed through their shadows. The B-form photographs achieve unprecedented clarity under her refined techniques. Franklin develops an intuition for which samples will yield the best results, selecting fibers with an artist's eye for quality. The work becomes almost mystical. She speaks of DNA wanting to reveal itself, of finding the conditions where the molecule feels comfortable enough to show its true structure. Franklin's mathematical analysis of the diffraction data reveals crucial measurements: the diameter of the DNA molecule, the spacing between its structural elements, and hints about its overall geometry. She fills pages with calculations, applying Bessel functions and Fourier transforms to extract three-dimensional information from two-dimensional patterns. The numbers begin to form a consistent picture. DNA measures 20 angstroms in diameter, with repeating units every 34 angstroms along its length. The precision amazes her. Nature has constructed this molecule with architectural exactitude. Franklin cross-checks every measurement, using different mathematical approaches to verify her results. The helical parameters emerge from the data like sculpture from marble, revealed through patient calculation rather than imposed by preconception. She begins to visualize DNA's structure with increasing clarity, though crucial details remain elusive. Working late into the night, she calculates and recalculates the angles and intensities in her X-ray photographs, building a precise three-dimensional picture of DNA's structure. The laboratory grows quiet as colleagues leave for home, but Franklin remains hunched over her light box, measuring diffraction spots with calipers and protractors. She develops severe eye strain from hours of peering at photographs, but continues working through the discomfort. Each calculation builds on previous results, creating an interconnected web of measurements that must all agree for the structure to be valid. Franklin checks her work obsessively, aware that a single error could propagate through subsequent calculations. The numbers dance in her dreams: helical pitches, molecular diameters, bond angles. She wakes with new insights, rushing to the laboratory to test midnight revelations against photographic evidence. Her laboratory notebooks fill with detailed measurements and theoretical calculations, creating a comprehensive database of DNA's physical properties. Future historians will marvel at these notebooks. Every experiment documented with exhaustive detail, every calculation shown in full, every observation recorded no matter how minor. Franklin's handwriting remains neat despite fatigue, her diagrams precise despite working by lamplight. She creates indices and cross-references, building a searchable archive of DNA knowledge. The notebooks reveal her thought process, hypotheses tested and rejected, patterns noticed and investigated, gradual convergence toward truth. She documents failures as thoroughly as successes, understanding that negative results also contribute to knowledge. The systematic record-keeping reflects her scientific philosophy. Progress comes through accumulation of facts, not flashes of inspiration. The most promising photographs emerge from her work with the B form of DNA, showing remarkably clear diffraction patterns that suggest a helical structure. One image in particular captivates her. The reflections form a nearly perfect cross, with additional spots creating a diamond pattern around the center. The symmetry speaks of underlying order, of a structure both simple and elegant. Franklin spends hours analyzing this photograph, extracting every possible measurement. The pattern confirms her growing conviction that DNA forms a helix, but questions remain about the number of chains and their arrangement. She prepares more samples, seeking to reproduce and improve upon this revealing image. The technical challenges multiply, maintaining perfect humidity, achieving optimal exposure times, developing plates without introducing artifacts. Yet Franklin persists, driven by the tantalizing glimpses of truth in her photographs. Franklin begins to suspect that DNA consists of two intertwined chains, but the exact arrangement remains elusive despite months of intensive investigation. The diffraction patterns show features that single helices cannot explain, absent reflections where single chains would produce spots, intensities that suggest multiple scattering centers. She constructs paper models, trying different configurations of two chains, but none perfectly match her experimental data. The phosphate groups must form the outside of the structure. Her evidence for this seems clear, but how do the bases fit inside? Franklin considers various possibilities: chains running in the same direction, chains running opposite, bases paired or unpaired. Each configuration would produce different diffraction patterns. She returns to her photographs, seeking the subtle clues that will distinguish between possibilities. She presents her findings at scientific seminars, describing DNA's helical nature and providing precise measurements of its dimensions, unaware that her words are being carefully noted by competitors. Franklin's presentations reflect her meticulous approach, dense with data, cautious in speculation, comprehensive in scope. She shows slide after slide of diffraction patterns, explaining how each contributes to understanding DNA's structure. The audience includes researchers from across Britain, many working on the same problem. They scribble notes as Franklin describes her two forms of DNA, her precise measurements, her evidence for helical structure. She answers questions thoroughly, sharing technical details about sample preparation and analysis methods. Franklin believes in scientific openness, unaware that some listeners view her presentations as intelligence-gathering opportunities rather than collegial exchange. The scientific community buzzes with excitement about DNA research as teams around the world race to solve the structure that will unlock the secret of genetic inheritance. Conferences crackle with competitive energy as researchers present partial results, probe competitors for information, form and break collaborative alliances. The stakes feel enormous. Whoever solves DNA's structure will achieve scientific immortality. Franklin observes the frenzy with mixed feelings. Part of her thrives on the competition, pushing herself harder, knowing others pursue the same goal. Yet, the political maneuvering disturbs her. Science should be about truth, not glory. She watches colleagues become secretive, paranoid, obsessed with priority. The collaborative spirit that attracted her to research seems poisoned by ambition. Franklin responds by redoubling her systematic approach, trusting that careful work will prevail over rushed speculation. Franklin's work attracts attention from Cambridge, where James Watson and Francis Crick are building theoretical models of DNA based on chemical principles and scattered experimental data. The young American Watson attends one of Franklin's seminars, struck by her crystallographic expertise and comprehensive data. He reports back to Crick, describing Franklin's measurements and her evidence for helical structure. The Cambridge duo work differently than Franklin. They build physical models based on chemical constraints, trying various configurations until one fits available data. They lack Franklin's experimental expertise, but compensate with theoretical boldness. Watson and Crick pump every visitor for information about DNA research elsewhere, assembling clues from multiple sources. Franklin's precise measurements provide crucial constraints for their model building, though she remains unaware of how her public presentations feed their efforts. Unbeknownst to Franklin, her seminar presentations and informal discussions provide crucial insights that help guide the Cambridge team's modeling efforts. Every measurement she shares, every technical detail she explains, becomes ammunition in the scientific race. Watson takes imperfect notes at her talk, misremembering some details but capturing enough to advance their work. The Cambridge team discusses Franklin's findings late into the night, incorporating her data into their evolving models. They speak of her with mixture of respect and frustration, admiring her experimental skill while dismissing her theoretical caution. The gender dynamics add another layer. Watson especially seems unable to see past Franklin's refusal to conform to feminine stereotypes. They nickname her Rosie, a diminutive she would despise, reducing her to a caricature rather than recognizing her as a formidable competitor. The pressure intensifies as Franklin realizes she's close to a breakthrough. But the final pieces of the structural puzzle remain tantalizingly out of reach. She can feel the solution hovering just beyond grasp, like a word on the tip of her tongue. The helical structure seems certain, the measurements consistent, yet the complete picture won't crystallize. Franklin pushes herself harder, working 18-hour days, neglecting meals and sleep. Her health begins to suffer, headaches from eye strain, backaches from hunching over equipment, persistent coughs from laboratory chemicals. Yet she cannot rest with the answer so close. She reviews old photographs with fresh eyes, recalculates measurements seeking overlooked clues, prepares new samples hoping for the perfect image that will resolve all ambiguities. The DNA molecule seems to taunt her, revealing just enough to prove progress possible while withholding final secrets. Her most famous photograph, later known as Photo 51, captures the B form of DNA with unprecedented clarity, showing the unmistakable X pattern that reveals the molecule's helical structure. The image emerges from the developer like a revelation, sharp, symmetrical, speaking eloquently of underlying order. Franklin stares at the pattern, recognizing immediately its significance. The cross shape confirms helical structure, while the diamond pattern provides precise measurements of the helix parameters. The photograph represents technical perfection, optimal humidity, precise beam alignment, exact exposure time, flawless development. All her months of refinement culminate in this single image. Franklin labels it carefully, adding it to her growing collection. Unaware she holds the key that will unlock DNA's secret. She begins the careful process of extracting every measurement from the photograph, building toward the complete structural solution that seems finally within reach. The image represents months of painstaking work, preparing perfect DNA samples, adjusting equipment settings, and timing expos. Photo 51 emerges from the developer like a revelation. Its crisp X pattern, providing the clearest evidence yet of DNA's helical structure and precise molecular dimensions. The darkroom's red light bathes Franklin's face as she lifts the dripping photograph from its chemical bath, her breath catching at the sight before her. The pattern spreads across the film with mathematical perfection, a bold cross at the center, surrounded by a diamond constellation of spots, each one positioned with nature's own precision. She has seen thousands of diffraction patterns, but this one speaks with unprecedented clarity. The image seems to pulse with significance, as if the DNA molecule itself has finally decided to reveal its deepest secret. Franklin's hands tremble slightly as she clips the photograph to the drying line, already calculating what measurements this extraordinary image will yield. Franklin stares at the photograph with growing excitement, recognizing that she holds in her hands the key to one of biology's greatest mysteries, the three-dimensional architecture of the genetic material. Years of preparation have led to this moment. Every failed experiment, every blurred photograph, every 16-hour day in the basement laboratory. The X pattern confirms what she has long suspected but could never quite prove. DNA forms a helix, its chains spiraling through space with geometric elegance. But Photo 51 offers more than confirmation. It provides a roadmap to the complete structure. The sharpness of the spots, the symmetry of the pattern, the precise angles between reflections, all speak of a molecule constructed with architectural perfection. Franklin feels the weight of discovery settling on her shoulders, the responsibility of being perhaps the only person on Earth who truly understands what this photograph reveals. The image reveals crucial details: the helical pitch, the diameter of the molecule, and the spacing between structural elements, all captured with mathematical precision in the diffraction pattern. Franklin measures each spot with calipers, noting positions to fractions of millimeters. The central cross indicates a helical structure with specific symmetry, not just any spiral, but one with precise geometric constraints. The layer lines tell her the helix repeats every 34 angstroms along its length. The systematic absences in the pattern reveal that the helix contains multiple chains, their arrangement creating interference patterns in the X-ray scattering. Even the relative intensities of different spots encode information about atomic positions within the molecule. Franklin's trained eye reads these abstract patterns like a musician reading a complex score, each feature contributing to the complete structural symphony. She begins intensive calculations based on Photo 51's data, working through complex crystallographic equations to extract every possible detail about DNA's structure. Franklin's desk disappears beneath layers of graph paper covered with mathematical derivations. She applies Bessel functions to analyze the helical diffraction, using techniques she mastered in Paris, but now pushing them to their limits. Each calculation builds on the previous one. The meridional reflections yield the rise per residue. The equatorial spots reveal the molecular diameter. The layer line spacing confirms the helical parameters. She cross-checks every result using different mathematical approaches, ensuring no errors creep into her analysis. The work requires total concentration. She disconnects her telephone and posts a sign on her door: "Do not disturb. Calculations in progress." Hours blur together as Franklin translates the photographic shadows into three-dimensional atomic coordinates. Franklin's analysis reveals that DNA consists of two chains running in opposite directions, with the phosphate groups forming the outer backbone of the double helix. The evidence emerges from careful study of the diffraction intensities. Certain reflections appear stronger than single-chain models would predict, while others are weaker. The pattern makes sense only if two chains wind around each other, their phosphate sugar backbones tracing parallel helices on the molecule's exterior. The anti-parallel arrangement explains subtle features in photo 51 that had puzzled her in earlier, less clear images. Franklin constructs paper models to visualize the structure, cutting out representations of the sugar and phosphate groups, arranging them according to her calculations. The model feels right, elegant, stable, with the charged phosphate groups positioned where they can interact with water molecules, explaining DNA's behavior in different humidity conditions. The mathematical precision of her measurements provides exact values for DNA's dimensions, a diameter of 2 nanometers, with the helical structure repeating every 3.4 nanometers along its length. These numbers emerge not from speculation, but from rigorous analysis of diffraction angles and intensities. Franklin fills pages with trigonometric calculations, converting the two-dimensional pattern into three-dimensional structure. The consistency amazes her. Measurements from different parts of the photograph all converge on the same values. She calculates that each turn of the helix contains 10 nucleotide units, creating a structure both compact and accessible. The dimensions suggest how DNA might pack into cell nuclei while remaining available for biological functions. Franklin begins to see how structure and function intertwine, the molecule's architecture perfectly suited for storing and transmitting genetic information. Working in methodical isolation, Franklin prepares a comprehensive manuscript describing her findings, determined to present a complete structural model supported by rigorous experimental evidence. She organizes her results with characteristic thoroughness. First the experimental methods, then the diffraction data, followed by mathematical analysis, and finally the structural conclusions. Each claim is supported by multiple lines of evidence. Franklin includes detailed diagrams showing how the diffraction pattern relates to molecular structure, making the complex mathematics accessible to biological readers. She writes and rewrites, ensuring every statement reflects the data accurately. The manuscript grows steadily, a monument to months of painstaking work. Franklin resists the urge to speculate beyond what her data supports, but she allows herself to hint at the biological implications of the anti-parallel double helix structure. Meanwhile, her strained relationship with Wilkins reaches a breaking point, leading to a decision that will have profound consequences for her career and scientific legacy. The tension has become unbearable. They schedule equipment use to avoid meeting, communicate only through written notes, maintain rigid territories within the shared laboratory space. Wilkins feels increasingly frustrated by Franklin's independence and her refusal to share preliminary results. From his perspective, they should be collaborating on the DNA problem. From hers, she has been hired to conduct independent research. The misunderstanding, rooted in poor communication from laboratory leadership, has poisoned their working relationship beyond repair. In a moment of profound frustration, Wilkins makes a choice that will reverberate through scientific history. He has maintained copies of some of Franklin's photographs, including her latest work, reasoning that as deputy director of the biophysics unit, he has the right to all data produced there. Unknown to Franklin, Wilkins shares photo 51 and her unpublished data with Watson and Crick at Cambridge, providing them with the crucial experimental evidence they need for their theoretical model. The meeting occurs casually. Watson visits King's College, hoping to mend relationships and perhaps establish collaboration. Wilkins, feeling isolated and frustrated, shows Watson the photograph almost impulsively, seeking validation for the unit's work. Watson stares at photo 51 with growing excitement, immediately grasping its significance. The clear helical pattern confirms the model he and Crick have been building, while the precise measurements allow them to adjust their structure to match reality. Wilkins also shares Franklin's unpublished measurements of DNA's dimensions, data she has presented in internal seminars but not yet published. Watson memorizes every detail, knowing this information will transform their speculative model into concrete reality. The Cambridge duo recognizes immediately that Franklin's photograph confirms their suspicions about DNA's helical structure. and provides the precise measurements needed to build an accurate model. Watson rushes back to Cambridge, burst into Crick's office with barely contained excitement. They have been building models based on chemical principles and fragmentary data, but Franklin's photograph provides the experimental foundation they desperately need. The X pattern speaks unambiguously of a helix, while the specific angles and spacings constrain their model building in crucial ways. They work through the night, adjusting their physical model to match Franklin's measurements. Metal plates representing bases, wire frameworks showing sugar-phosphate chains, all must be repositioned to match the experimental reality captured in Photo 51. The model that emerges feels inevitable, as if Franklin's data has guided their hands to the only possible solution. Watson and Crick work feverishly to incorporate Franklin's data into their theoretical framework, racing to publish their findings before she can complete her own comprehensive analysis. They know others are close to the solution. Linus Pauling in California has published an incorrect three-chain model, but he will surely recognize his error soon. Franklin herself must be near the complete structure, given the quality of her data. The Cambridge pair works with desperate intensity, checking their model against every piece of available information. They consult chemistry textbooks to ensure bond angles are reasonable, calculate whether their proposed base pairs fit within Franklin's measured diameter, verify that their structure produces the diffraction pattern she observed. The model must be not just consistent with her data, but chemically and biologically plausible. Days blur together in a frenzy of construction, calculation, and revision. Franklin continues her meticulous work, unaware that her most important discoveries are being used to construct the very model she herself is working toward. In her basement laboratory, she refines her analysis of Photo 51, extracting ever more precise measurements. She prepares new samples, seeking to capture equally revealing photographs of DNA's A form to complete her structural study. Franklin's notebooks fill with calculations as she builds toward a complete three-dimensional model. She considers how the bases might fit within the helical framework, sketching possible arrangements. The anti-parallel chains create a puzzle. How do the bases bridge between them? Franklin methodically works through possibilities, checking each against her diffraction data. She feels close to the complete solution, but maintains her disciplined approach, refusing to publish until every detail is confirmed. She refines her understanding of DNA's structure, determining that the two helical chains are held together by hydrogen bonds between complementary base pairs. The insight comes gradually as Franklin analyzes the density distribution within DNA revealed by her X-ray data. The interior of the helix shows regular features suggesting ordered base stacking. She calculates the space available between the anti-parallel chains, just enough for the flat bases to fit, oriented perpendicular to the helical axis. But how do they connect? Franklin's chemical knowledge suggests hydrogen bonding, the same weak forces that hold water molecules together. She begins to work out which bases might pair with each other, constrained by the uniform diameter her photographs reveal. The large purines must pair with smaller pyrimidines to maintain consistent helix width. The realization dawns slowly: adenine with thymine, guanine with cytosine, held together by precise hydrogen bond geometry. The realization dawns that DNA's structure perfectly explains its biological function. The complementary base pairing provides a mechanism for accurate replication of genetic information. Franklin sits back from her calculations, struck by the elegant simplicity of nature's solution. If bases pair specifically, then each strand contains the information needed to reconstruct its partner. During cell division, the strands could separate, each serving as a template for creating a new complementary strand. The structure itself embodies the mechanism of heredity. She thinks of all the biological puzzles this might solve: how genetic information passes from parent to offspring, how cells maintain their identity through countless divisions, how life achieves such remarkable fidelity in transmitting biological instructions. The molecular architecture reveals not just what DNA looks like, but why it looks that way. Franklin prepares detailed drawings and calculations showing how the 4 DNA bases, adenine, thymine, guanine, and cytosine, pair specifically with each other within the double helix. Her artistic skills, developed through years of crystallographic work, allow her to create clear, accurate representations of the molecular structure. She draws the bases with their correct chemical structures, showing how hydrogen bonds form between complementary pairs. The drawings reveal the beautiful symmetry of the pairing scheme, adenine's double ring structure fitting perfectly with thymine's single ring, guanine's triple hydrogen bonding complementing cytosine's acceptor sites. Franklin calculates bond angles and distances, ensuring her drawings reflect physical reality. The illustrations grow increasingly sophisticated as she adds the sugar phosphate backbone, showing how the bases stack like steps in a spiral staircase. Her notebooks reveal the depth of her understanding. She has not only determined DNA's structure, but also grasped its profound implications for heredity and reproduction. Franklin writes extensive notes exploring how the double helix might function in living cells. She considers how enzymes might unwind the helix for replication, how the complementary base pairing ensures accurate copying, how mutations might arise from pairing errors. Her insights extend beyond structure to mechanism, beyond static architecture to dynamic function. Franklin sketches possible schemes for DNA replication, anticipating discoveries that won't be confirmed for years. She understands that she stands at the threshold of a new era in biology, where molecular structure illuminates the fundamental processes of life. The knowledge feels almost too large to contain, demanding to be shared with the scientific world. The scientific community remains unaware of Franklin's progress as she works toward a complete publication that will establish her priority in solving DNA's structure. She maintains her characteristic discretion, sharing results only in internal seminars and avoiding premature announcements. Franklin believes in the power of comprehensive publication. Better to wait and present complete findings than rush incomplete work into print. She continues refining her manuscript, adding new experimental evidence, strengthening her mathematical arguments, preparing figures that clearly convey the double helix structure. The work proceeds steadily, but without haste. Franklin has no idea that competitors armed with her own data race toward publication. She works in isolation, trusting that careful science will ultimately prevail over rushed speculation. Watson and Crick submit their famous paper to Nature, describing the double helix model and acknowledging Franklin's X-ray data only in passing. Their manuscript arrives at the journal in early April 1953, a mere 2 pages that will transform biology. The brevity masks the profound implications. They describe the anti-parallel double helix, the complementary base pairing, and hint at the replication mechanism with masterful understatement. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The acknowledgments mention Franklin and Wilkins briefly, noting that the authors were stimulated by unpublished experimental results and ideas from King's College. The passive construction obscures the crucial role of Franklin's Photo 51 in confirming their model. The publication of the Watson-Crick model in April 1953 sends shockwaves through the scientific world, establishing them as the discoverers of DNA's structure. The paper appears in Nature on April 25th, accompanied by supporting articles from Wilkins and Franklin that provide experimental evidence for the proposed structure. Scientists worldwide immediately grasped the significance. The double helix explains so much that had been mysterious about heredity and biological information. The elegant simplicity of complementary base pairing captures imaginations, suggesting mechanisms for replication and mutation that researchers rush to investigate. Watson and Crick become overnight celebrities in the scientific community, their names forever linked with one of biology's greatest discoveries. The model they propose, built on Franklin's experimental foundation, launches the molecular biology revolution. Franklin learns of the Cambridge publication with a mixture of recognition and disappointment. Her data has been crucial to the discovery, but her contribution is minimized. She reads the Watson-Crick paper with professional interest, recognizing immediately that their model matches her own developing understanding. The structure they propose agrees perfectly with her diffraction data, confirming her own calculations. Yet something feels wrong about the sequence of events. How did they achieve such precise measurements without extensive crystallographic work? The brief acknowledgement of King's College contributions seems inadequate given that Photo 51's parameters define their model. Franklin maintains her professional composure, but close friends notice her subdued mood. She has worked so hard, come so close, only to see others claim the prize using her own experimental evidence. She publishes her own detailed analysis of DNA's structure in the same issue of Nature, providing the experimental foundation that supports the Watson-Crick model. Franklin's paper presents the crystallographic evidence with characteristic thoroughness, precise measurements, careful analysis, rigorous mathematical treatment. She describes both A and B forms of DNA, showing how water content affects the molecular structure. Her data confirms the double helix model while providing details the theoretical paper lacks. Franklin's figures include actual X-ray photographs and detailed measurements that future researchers will use to refine understanding of DNA structure. The paper stands as a monument to experimental excellence, even as it appears to play a supporting role to the more famous theoretical model. Readers who examine both papers carefully recognize that Franklin's data made the Watson-Crick model possible. Franklin's paper offers rigorous proof of the double helix structure, with precise measurements and crystallographic analysis that validate the theoretical model. Where Watson and Crick provide elegant speculation, Franklin delivers hard data. Her measurements of helical parameters, pitch, diameter, repeat distance, define the quantitative framework within which all future DNA research will operate. She shows how different humidity conditions affect DNA structure, explaining observations that had confused earlier researchers. The mathematical analysis demonstrates mastery of crystallographic theory, extracting three-dimensional information from two-dimensional patterns with unprecedented precision. Franklin's paper becomes the experimental bedrock on which the DNA revolution builds, even as history initially celebrates the theoretical insight more than the experimental achievement. Franklin's paper offers rigorous proof of the double helix structure, with precise measurements and crystallographic analysis that validate the theoretical model. Where Watson and Crick provide elegant speculation, Franklin delivers hard data. Her measurements of helical parameters, pitch, diameter, repeat distance, define the quantitative framework within which all future DNA research will operate. She shows how different humidity conditions affect DNA structure, explaining observations that had confused earlier researchers. The mathematical analysis demonstrates mastery of crystallographic theory, extracting three-dimensional information from two-dimensional patterns with unprecedented precision. Franklin's paper becomes the experimental bedrock on which the DNA revolution builds, even as history initially celebrates the theoretical insight more than the experimental achievement. The scientific community celebrates the solution of DNA's structure as one of biology's greatest achievements, launching the molecular revolution that will transform medicine and genetics. Conferences buzz with excitement as researchers grasp the implications of the double helix. The complementary base pairing suggests immediate experiments. How does DNA replicate? How do mutations arise? How does genetic information translate into proteins? Funding pours into molecular biology as governments and foundations recognize the field's potential. Young scientists abandon traditional biology for this new molecular approach, seeking to understand life at its most fundamental level. The double helix becomes an icon, its spiral form appearing on journal covers, in textbooks, eventually in popular culture. The revolution Franklin helped launch proceeds with breathtaking speed. Franklin's role in the discovery becomes a subject of historical debate, with many recognizing that her experimental work provided the crucial evidence for understanding DNA's architecture. As years pass, historians of science piece together the complex story of DNA's discovery. They find Franklin's notebooks, study her photographs, interview surviving participants. The picture that emerges shows her central role in providing the experimental foundation for the double helix model. Photo 51 becomes famous as the key evidence that confirmed Watson and Crick's structure. Scholars debate whether Franklin would have reached the complete structure independently. Franklin's role in the discovery becomes a subject of historical debate, with many recognizing that her experimental work provided the crucial evidence for understanding DNA's architecture. As years pass, historians of science piece together the complex story of DNA's discovery. They find Franklin's notebooks, study her photographs, interview surviving participants. The picture that emerges shows her central role in providing the experimental foundation for the double helix model. Photo 51 becomes famous as the key evidence that confirmed Watson and Crick's structure. Scholars debate whether Franklin would have reached the complete structure independently. The publication of DNA's structure marks the beginning of a new era in biology, as scientists around the world grasp the profound implications of the double helix discovery. In laboratories from London to Tokyo, researchers pin up diagrams of the elegant spiral structure, marveling at nature's solution to the puzzle of heredity. The complementary base pairing, adenine with thymine, guanine with cytosine, speaks of a mechanism so simple yet so powerful that it takes collective breath away. Graduate students abandon their previous research topics, drawn to this new molecular frontier where life's deepest secrets seem suddenly accessible. The structure itself becomes a kind of Rosetta Stone, allowing scientists to decode biological processes that had remained mysterious since the dawn of scientific inquiry. Conference halls buzz with excited discussions about replication mechanisms, mutation patterns, and the molecular basis of evolution itself. Franklin watches as the scientific community celebrates Watson and Crick's achievement, knowing that her X-ray crystallography provided the experimental foundation for their theoretical model. She attends seminars where speakers project her Photo 51 on screens, explaining how its clear X pattern confirmed the helical structure. The image has become iconic almost overnight, yet few mention the months of painstaking work required to capture it. Franklin maintains her professional composure, congratulating the Cambridge team on their insight, while continuing her own research. In quiet moments, she reflects on the strange path science sometimes takes, how her methodical experimental work enabled others' theoretical leap. The experience reinforces her belief in the importance of rigorous data collection, even as she recognizes that scientific glory often goes to those who synthesize rather than those who discover. Despite the complex circumstances surrounding the DNA discovery, Franklin chooses to move forward, rather than dwell on questions of credit and recognition. She could spend energy fighting for acknowledgement, demanding proper attribution for her crucial role. Legal battles over scientific priority have destroyed careers and friendships throughout history. But Franklin possesses something more valuable than pride, an unquenchable curiosity about the natural world. New questions beckon, new structures await discovery. She channels any frustration into determination, resolving to make her next contributions so significant that proper recognition becomes inevitable. Friends admire her grace under circumstances that would embitter lesser scientists. Franklin's notebooks from this period show no self-pity, only plans for future experiments, ideas for new crystallographic techniques, sketches of molecular structures yet to be solved. She accepts a position at Birkbeck College, where she begins groundbreaking research on the structure of RNA and viruses, applying her crystallographic expertise to new biological mysteries. The laboratory at Birkbeck offers a fresh start, free from the poisoned atmosphere at King's College. Professor J.D. Bernal, a pioneer in biological crystallography, welcomes Franklin with genuine respect for her abilities. She sets up her X-ray equipment with practiced efficiency, eager to tackle problems even more challenging than DNA. RNA, the molecule that carries genetic information from DNA to protein synthesis, presents unique difficulties. It's less stable than DNA, harder to crystallize, more prone to degradation. Viruses offer their own challenges. These strange entities at the border between living and non-living. their protein coats protecting nucleic acid cores. Franklin approaches these puzzles with renewed energy, her expertise now recognized and valued by colleagues. The DNA discovery unleashes a revolution in molecular biology, as researchers begin to understand how genetic information is stored, replicated, and transmitted between generations. The double helix structure provides a conceptual framework that organizes previously scattered observations into coherent understanding. Scientists realize that DNA's sequence of bases constitutes a code, a four-letter alphabet spelling out the instructions for building and maintaining living organisms. The anti-parallel strands explain how cells can copy DNA with such fidelity, each strand serving as a template for its partner. Mutations, previously mysterious changes in heredity, now make sense as alterations in base sequence. Evolution itself gains molecular explanation, natural selection acting on variations in DNA sequence. The implications ripple outward, touching every branch of biology from development to disease. Franklin's precise measurements of DNA's dimensions become the foundation for countless future studies, providing the structural framework for understanding genetic mechanisms. Every researcher investigating DNA's function relies on the parameters she established: the 34 angstrom helical repeat, the 20 angstrom diameter, the 3.4 angstrom rise per base pair. These numbers, extracted from shadows on photographic plates through mathematical analysis, define the physical constraints within which all genetic processes operate. Enzymes that replicate DNA must accommodate these dimensions. Proteins that regulate gene expression must recognize this structure. Even decades later, as technology advances to allow direct visualization of DNA molecules, Franklin's measurements prove remarkably accurate. Her experimental rigor established benchmarks that anchor an entire field of study. Medical researchers recognize that understanding DNA's structure opens unprecedented opportunities for diagnosing and treating genetic diseases. Sickle cell anemia, one of the first diseases understood at a molecular level, results from a single base change in the DNA sequence coding for hemoglobin. This realization that inherited diseases stem from specific alterations in DNA structure transforms medical thinking. Physicians begin to envision treatments that address causes rather than symptoms. Genetic counselors can explain inheritance patterns using the double helix model, showing how traits pass from parents to children through complementary base pairing. Research hospitals establish molecular medicine departments, recruiting scientists trained in the new biology. The dream of reading individual DNA sequences, seemingly impossible in 1953, becomes a driving goal that will eventually revolutionize personalized medicine. The pharmaceutical industry begins investing heavily in molecular biology research, recognizing that DNA's structure provides a roadmap for developing new therapeutic approaches. Drug companies hire crystallographers, molecular biologists, and biochemists, building research facilities focused on understanding disease at the molecular level. The idea emerges that drugs might target specific DNA sequences, or the proteins they encode. Antibiotics could be designed to interfere with bacterial DNA replication, while leaving human cells unharmed. Cancer drugs might block the uncontrolled DNA copying that characterizes malignant growth. The double helix structure suggests strategies for antiviral medications, compounds that prevent viruses from hijacking cellular DNA machinery. Investment flows into universities and research institutes, funding the basic science that will yield tomorrow's treatments. Franklin's work on virus structures at Birkbeck reveals the elegant simplicity of these molecular machines, showing how they hijack cellular machinery for reproduction. Her X-ray photographs capture viruses in crystalline arrays, their geometric perfection belying their devastating biological effects. The tobacco mosaic virus, her first major target, appears as a hollow cylinder with RNA spiraling inside a protective protein coat. Franklin's images show how viral proteins assemble with mathematical precision, creating containers perfectly sized for their genetic cargo. She discovers that slight environmental changes cause dramatic structural shifts, explaining how viruses inject their genetic material into host cells. Each photograph requires weeks of preparation, growing virus crystals, maintaining them in precise conditions, capturing diffraction patterns before inevitable degradation. The work demands all her crystallographic skill, plus new techniques for handling biological samples. Her studies of tobacco mosaic virus and poliovirus provide crucial insights into viral structure and function, contributing to the development of vaccines and antiviral treatments. Franklin's structural data reveals vulnerable points in viral architecture, sites where drugs might interfere with assembly or host cell recognition. Her work on poliovirus proves particularly significant as the world battles polio epidemics. She shows how the virus's protein shell protects its RNA core, while providing mechanisms for cellular attachment and entry. This structural understanding guides vaccine developers, helping them create weakened viruses that stimulate immunity without causing disease. Franklin collaborates with virologists and immunologists, her structural insights complementing their biological studies. The interdisciplinary approach, still novel in the 1950s, establishes patterns for modern biomedical research. The techniques Franklin pioneered for X-ray crystallography of biological molecules become standard tools in laboratories worldwide, enabling countless structural discoveries. Her methods for maintaining biological samples during long X-ray exposures, her mathematical approaches to analyzing complex diffraction patterns, her systematic documentation of experimental conditions all become part of crystallography's standard practice. Graduate students learn Franklin's rules for preparing nucleic acid fibers. Crystallography textbooks cite her innovations in humidity control and sample mounting. The precision she brought to biological crystallography raises standards throughout the field. Researchers attempting to solve new structures study her notebooks, learning not just techniques but an approach to experimental science that values thoroughness over speed, accuracy over speculation. Young scientists inspired by the DNA breakthrough flock to molecular biology programs, launching careers that will further expand our understanding of life's molecular mechanisms. Universities scramble to establish molecular biology departments, hiring faculty who can teach this new science. Students who might have pursued traditional biology or chemistry find themselves drawn to this interdisciplinary field where physics, chemistry, and biology merge. They learn to think in molecular terms, visualizing cellular processes as interactions between three-dimensional structures. Franklin becomes a role model for female scientists, demonstrating that women can make fundamental contributions despite institutional barriers. Her example inspires a generation of women to pursue careers in crystallography and structural biology, slowly changing the face of science. Franklin's meticulous approach to experimental science becomes a model for rigorous research, emphasizing the importance of comprehensive data collection before drawing conclusions. Her notebooks, studied by historians and scientists alike, reveal a mind that refused shortcuts or premature speculation. Every experiment is documented with exhaustive detail: temperature, humidity, exposure time, development conditions. Failed experiments receive as much attention as successes, their lessons carefully analyzed. This systematic approach, sometimes criticized as overly cautious during her lifetime, proves its worth as structural biology matures. Researchers attempting to reproduce or extend her work find they can follow her protocols exactly, achieving consistent results. The reliability of her data allows future scientists to build confidently on her foundations. The double helix structure explains the mechanism of genetic inheritance with elegant simplicity. Each strand serves as a template for creating its complement during cell division. This copying mechanism, implicit in the complementary base pairing Franklin helped establish, revolutionizes understanding of cellular reproduction. Scientists quickly confirm that DNA strands separate during replication, each serving as a template for synthesizing a new partner strand. The process ensures that genetic information passes faithfully from cell to cell, from parent to offspring. Errors in copying explain mutations, while repair mechanisms that correct mismatched bases maintain genetic stability. The structural insight Franklin provided transforms abstract concepts of heredity, into concrete molecular processes that can be studied, manipulated, and eventually harnessed for human benefit. Researchers begin to decode the genetic code itself, determining how the sequence of DNA bases specifies the amino acid sequence of proteins. The double helix structure provides the physical framework for understanding how linear information in DNA becomes three-dimensional proteins that perform cellular functions. Scientists discover that three base sequences code for specific amino acids, creating a universal language of life. This genetic code, conserved across all organisms from bacteria to humans, confirms the fundamental unity of life on Earth. Franklin's structural work made this decoding possible by establishing how information could be stored in DNA's linear sequence while remaining accessible for cellular machinery to read and translate. The discovery leads to the development of genetic engineering techniques, allowing scientists to manipulate DNA sequences and create modified organisms. Understanding DNA's structure enables researchers to cut and splice genetic material with molecular precision. Restriction enzymes that recognize specific DNA sequences become molecular scissors. DNA ligases that join fragments become molecular glue. Scientists learn to insert genes from one organism into another, creating bacteria that produce human insulin, plants resistant to pests, and research animals that model human diseases. Each advance builds on the structural foundation Franklin helped establish, her precise measurements defining the molecular landscape within which genetic engineers operate. Franklin's health begins to decline due to her extensive work with X-rays, the radiation exposure from years of crystallographic research taking its toll on her body. The safety protocols of the 1950s, primitive by modern standards, offered little protection from the cumulative effects of radiation. Franklin, like many crystallographers of her generation, absorbed significant doses during long hours of equipment adjustment and sample positioning. She begins experiencing fatigue and abdominal pains, but initially attributes them to overwork. Medical examinations reveal ovarian cancer, likely caused by radiation damage to rapidly dividing cells. The diagnosis devastates her colleagues, who recognize the cruel irony. The same X-rays that revealed life's molecular secrets now threaten the life of one who wielded them most skillfully. Even as illness weakens her, Franklin continues her research with characteristic determination, publishing important findings on virus structure until shortly before her death. She schedules chemotherapy treatments around experiments, refusing to abandon ongoing projects. Her research group at Birkbeck marvels at her resilience, watching her direct experiments from a wheelchair when standing becomes too difficult. Franklin completes major papers on tobacco mosaic virus structure, her handwriting remaining precise despite trembling hands. She mentors younger scientists, passing on not just technical knowledge, but her philosophy of meticulous experimentation. Hospital stays become working sessions where she analyzes data and plans future experiments, her mind sharp even as her body fails. She passes away in 1958 at age 37. Her life cut short by ovarian cancer, likely caused by radiation exposure from her groundbreaking X-ray crystallography work. Franklin faces death with the same clear-eyed determination. She brought to scientific problems. She makes arrangements for her research to continue, ensuring her data and samples pass to capable hands. Her final papers appear posthumously, their elegant prose and rigorous analysis showing no hint of the pain she endured while writing them. Colleagues gather for memorial services in London and Paris, sharing memories of a scientist whose dedication to truth surpassed personal ambition. They speak of her kindness to students, her generosity with technical advice, her unwavering commitment to experimental excellence. The Nobel Prize in Physiology or Medicine is awarded to Watson, Crick, and Wilkins in 1962 for the discovery of DNA's structure, with Franklin's contribution acknowledged but not honored. Nobel rules prohibit posthumous awards, preventing formal recognition of Franklin's crucial role. Watson's acceptance speech mentions her contributions briefly, while Wilkins acknowledges the importance of X-ray crystallography without naming Franklin specifically. The omission strikes many as unjust, particularly those who understand how Photo 51 enabled the theoretical breakthrough. Debates emerge about scientific credit and the treatment of women in science. Some argue Franklin would have shared the prize had she lived. Others question whether the Nobel Committee would have recognized a woman's contributions in that era. Franklin's scientific legacy grows over time, as historians and scientists recognize the crucial importance of her experimental work in solving DNA's structure. Biographers uncover the full story of Photo 51 and its journey from Franklin's laboratory to Watson and Crick's model. Her notebooks reveal how close she came to independently solving the complete structure. Scientists reviewing her work marvel at its quality and thoroughness, recognizing that modern structural biology rests on foundations she helped establish. Museums display her photographs and equipment, teaching new generations about the woman whose X-ray vision revealed life's blueprint. Franklin becomes an icon for women in science, her story inspiring those who face similar barriers. Her precise measurements and rigorous experimental approach become textbook examples of how careful science provides the foundation for theoretical breakthroughs. Crystallography courses use Franklin's work to demonstrate proper technique and data analysis. Science historians cite her research as exemplifying the crucial but often undervalued role of experimental science in major discoveries. Her approach, methodical, thorough, skeptical of premature conclusions, offers timeless lessons for researchers in any field. Students learn that breakthrough discoveries often depend on painstaking experimental work, performed with little recognition or glory. Franklin's legacy teaches that science advances through the patient accumulation of reliable data, as much as through flashes of theoretical insight. The molecular biology revolution she helped launch transforms medicine, agriculture, and biotechnology, touching virtually every aspect of human life. Genetic testing identifies disease risks before symptoms appear. Gene therapy offers hope for previously incurable conditions. Agricultural scientists develop crops that feed billions while using fewer resources. Forensic investigators solve crimes through DNA evidence. Evolutionary biologists trace human origins through genetic sequences. Each application builds on the structural understanding Franklin provided, her shadow-captured images echoing through decades of scientific progress. The double helix she helped reveal becomes humanity's tool for understanding and shaping life itself. Modern genetic technologies, from DNA sequencing to gene therapy, all trace their origins back to the structural understanding Franklin's work helped provide. The Human Genome Project, reading all 3 billion letters of human DNA, depends on knowing how those letters arrange in space. CRISPR gene editing, precisely altering DNA sequences, requires understanding the double helix structure Franklin illuminated. Personalized medicine, tailoring treatments to individual genetic profiles, builds on her foundation. Even as technology advances beyond anything Franklin could have imagined, her contributions remain fundamental. Every genetic breakthrough carries within it the echo of those long nights in the King's College basement, where a determined woman coaxed secrets from shadows, revealing the architecture of life itself. The X-ray photographs she captured with such patience and skill continue to light the way forward, guiding humanity toward ever deeper understanding of our molecular nature. The woman who captured shadows understood something profound about the nature of discovery. The truth reveals itself not to those who demand quick answers, but to those patient enough to listen to what the invisible world has to say. Rosalind Franklin's journey from a curious child mixing chemicals in her nursery to the scientist who photographed DNA's secret architecture speaks to something deeper than scientific achievement. It whispers of dedication that transcends recognition, of contributions that echo through generations, even when the contributor's name fades from memory. Her X-ray photographs didn't just reveal molecular structures. They opened a window into the very essence of what makes us human, what connects us to every living thing that has ever existed on this planet. In those ghostly diffraction patterns captured through years of radiation exposure that would ultimately claim her life, Franklin gave humanity its first clear view of the blueprint that writes our existence. As we've traced her path through basement laboratories, and dark rooms filled with chemical scents, we've witnessed more than the solving of a scientific puzzle. We've seen how one person's unwavering commitment to truth can illuminate pathways for all who follow. Franklin's meticulous measurements, the 34 angstrom repeat, the 20 angstrom diameter, the precise angles of diffraction spots, became the foundation upon which modern biology builds its understanding. Every genetic test that reveals disease risk, every treatment that targets faulty genes, every crop that feeds hungry populations carries within it the legacy of those long nights when she coaxed secrets from crystallized DNA fibers. The double helix she helped reveal has become humanity's tool for reading and eventually rewriting the book of life itself. What strikes us most deeply isn't the scientific triumph, but the human grace with which Franklin faced both discovery and disappointment. When others claimed recognition for insights built on her experimental foundation, she turned not to bitterness, but to new questions, new structures waiting to be revealed. Her work on viruses, pursued even as cancer weakened her body, showed that true scientists seek knowledge for its own sake, not for glory or prizes. This dedication to truth over recognition offers a different kind of lesson, that our contributions to human understanding matter more than whether our names appear in headlines. Tonight, as sleep approaches and your mind settles into quietness, remember that somewhere in laboratories around the world, researchers still use techniques Franklin pioneered, still reference measurements she established, still build upon foundations she laid with such care. The X-ray vision she developed didn't just peer into molecular structures, it illuminated a path forward for all who seek to understand life's deepest mysteries. In every strand of DNA in your body, in every cell dividing and copying its genetic instructions with remarkable fidelity, lives the echo of Rosalind Franklin's patient work. Let her story remind you that the most profound discoveries often emerge not from flashes of inspiration, but from quiet persistence, from showing up day after day to do careful work, even when recognition seems unlikely. As you drift toward peaceful rest, carry with you the image of a determined woman in a dimly lit laboratory, transforming shadows into knowledge, revealing through patient observation what others could only imagine. In that transformation lies a truth that transcends science, that dedication, precision, and quiet courage can unveil wonders that change the world, even if the world doesn't always remember to say thank you. Sleep well, knowing that in the architecture of your very being lives the legacy of Rosalind Franklin, the woman whose steady hands and clear vision first showed us the elegant spiral at the heart of life itself.